Science in marine and atmospheric science at the University of Miami, where he worked with Dr. Peter Glynn on marine invertebrate ecology and physiology, as well as invasion ecology. And then he moved on to the PhD program in ecology, behavior, and evolution at the University of California, San Diego, where he's working in Dr. Jonathan Shurin's aquatic ecology laboratory. So Josh has already been quite successful, achieving multiple awards, including the National Science Foundation Graduate Research Fellowship, among others. And of course, the reason he's here today, the SSE Rosemary Grant Award. His dissertation focuses on how host-associated and free-living microbial communities are shaped by and influence host ecology, adaptation, and ecosystem function. And he'll be talking to us today about unraveling the causes and consequences of microbiome variation in freshwater ecosystems. So, Josh, welcome and take it away.
Thank you so much for the very kind introduction, Amber, and thank you to SSE for the award and for giving me this opportunity to present my work to you all. And like Amber just mentioned, today we're talking about sort of my dissertation work and really sort of the broad underlying thing I'm trying to get out with my dissertation work is really sort of unraveling the causes and consequences of microbiome variation in freshwater ecosystems. So just want to start out by sort of mentioning how microbiomes shape host ecology and evolution. And so what I have here is a figure of a Daphnia, which is a zooplankton in freshwater ecosystems, and all its different associated microbes. And these different microbes can shape hosts in various ways. For example, we have a good idea that gut immune function and gut physiology in general is highly modulated by the microbiome. And these sort of effects can also sort of scale up and has been implicated in how hosts respond to changes in the environment. And these sort of changes in the hosts could sort of scale up to influence higher order processes like host population dynamics over time, or even potentially, which I'm interested in thinking about, is how these could affect sort of in indirect ways how ecosystems function. But sort of a central problem in understanding these pretty essential communities is that there's considerable variation and plasticity within and across hosts. For example, we could think about how there's individual variation within a single host where one's microbiome could change over time or over different environments. Or another example could be sort of intraspecific variation. So sort of two individuals of the same species sort of having highly differentiated microbiomes. And then another level of potential variation is sort of interspecific variation, which I'll be talking about a good amount during this talk. And it's really sort of another sort of mechanism for variation in these communities where we see sort of highly varying communities between two individuals of different species. And so really it's important to understand this variation because these communities can influence the ecology and evolution of hosts and could be important to sort of understand how organisms are responding to changes in our environment. So how can we better understand this variation? And sort of a framework that I want to introduce and sort of reference for the rest of this talk is really sort of conceptualizing hosts as meta communities. And I know I'm giving a talk to Evolution Society, so this is a lot of eco stuff, but I think sort of understanding this meta community approach is important. And really what we think about is sort of how we can imagine an individual host as being an ecosystem with its own microbiome. And there's sort of this pool of microbes in the environment and sort of across all hosts. And we get sort of dispersal of the species pool between hosts and the environment. And importantly, there's sort of this thought that sort of this interaction between hosts and their environment and sort of dispersal of microbes between hosts and the environment could sort of shape the phenotype of the hosts. And so in this figure here, the noses of these mice have different colors, sort of denoting that their phenotypes could be changing as a function of this dispersal and these microbiomes. And so what I want you to introduce is sort of this feedback where sort of there's this host by microbiome by environment feedback where hosts are interacting with one another and they're interacting with their microbiome, and then that's sort of shaped by what's in the environment. And the composition of these communities are sort of determined by these four basic processes that are sort of what we think about in community ecology and are sort of a close, similar sort of framework to evolution were really the sort of dispersal, selection, drift, and speciation shaping these communities. So with this sort of framework in mind, I want to try to understand and unravel what's sort of driving variation in these communities. So what are the overarching questions that sort of underlie everything in my work? One being sort of like trying to identify the eco-evo causes or the processes that shape microbiome variation. And then the second question, which is considerably harder to get to, and I'm going to be talking about this a little bit, sort of how we could think about the potential causes of this variation for sort of host fitness and ecosystem function. So I've just laid out a very broad sort of framework that I'll be thinking about and working in, but I'll be talking about some very specific research questions that I'll lay out now. One, I'll be sort of talking about a project in my dissertation, which asks how host-associated and free-living microbiomes, whether they have divergent diversity and assembly patterns across gradients. And I'll be sort of comparing the gut microbiomes of fish and the microbiomes we find in lake environments and sort of what processes shape variation in their community structure. The second project I'll be talking about sort of asks the question of how host community diversity and composition affects microbiome. So this is thinking about sort of interactions between hosts and how that could shape their microbiomes of individual hosts and also the environment. So this sort of is connected to that host by microbiome by environment feedback. And this is an experiment I did by manipulating the composition and diversity of zooplankton communities and experimental mesocosms. And then finally, I'll be talking about my proposal to SSE for this fellowship, which is really asking this question of sort of what are the consequences of microbiome divergence between hosts for host fitness and ecosystem function. And to be sort of getting at this question, I'll be using sort of this benthic limnetic species pair divergence in stickleback. That is a well-known model in evolutionary biology. Sweet. So let's get after this first question. Do host-associated and free-living microbes have divergent diversity and assembly patterns across gradients? And so these are my great co-authors on this project. And really what this project is about is sort of comparing microbiomes between free-living, in this case, lake water microbes, and host-associated. So gut microbiomes of fish in two different systems. So we have lakes in the Sierra Nevadas, sort of mountain alpine lakes, and the gut microbiome of brook trout. And then another parallel system with Vancouver Island lakes and three-spine stickleback gut microbiomes. So free-living host-associated communities are distinct, but we once thought about microbes as being really just this part of food webs that was solely responsible for biogeochemical cycling. So what I'm showing you here is a figure of an aquatic food web from like the 1940s. And what I want to point out is they once thought that there's this bacterial ooze that was only sort of involved in biogeochemical cycling. But as I mentioned earlier, microbes can sort of form these really intimate relationships with hosts. And we don't really know whether these different communities are shaped by different processes and have different patterns across space and time. And so this sort of question is whether these communities exhibit divergent diversity and assembly patterns across gradients. So what did we do to get at this question? Like I mentioned, we went and sampled brook trout from 15 lakes in Sierra Nevadas in California. And we took fecal samples from a bunch of fish. And then we also sort of did this in parallel. This is from Diana Rennison's lab, led by Andy Herrera. And what they did is they sampled stickleback from 20 lakes, and then also sampled lake water from the same lakes as well. And so we sort of saw this parallel study design and thought it would be good to sort of put all of our data together to answer this sort of broader question. Sweet. So yeah, exactly what I just said. So we have to establish, okay, are these communities even different in the first place? What we can see is, yeah, they're highly divergent. And so what I'm showing you here is a PCOA plot of microbiomes in either fish or lake water. Triangles are sort of representative of fish. Circles are representative of lake water. And the colors denote the different systems. So blue is for the Sierras, orange is for Vancouver. And that'll be sort of the color scheme for the rest of this talk. And what we can see is that there's this divergence sort of along the first axis between fish and lake water samples. And so you think that maybe there would be a lot of overlap between these two since fish are sort of in this soup of microbes in the water, but we actually see this strong distinction within and across systems. So let's get at some specific questions that we're going to answer in this project. So first, do they have varying diversity? So this will give us an idea of like the different processes that are shaping their microbiomes. Two, whether they display different distance decay rates, and this is sort of a fundamental biogeographic pattern that's well known and I'll be sort of talking about. And three, sort of asking what is the contribution of neutral forces, so sort of random stochastic processes in shaping these communities. And this is really important because understanding what's shaping these communities could be important for host ecology and evolution and also sort of ecosystem functioning. So do they have varying microbial diversity? And when I talk about microbial diversity, I'll be talking about alpha diversity, which is sort of index of how many species and or the relative abundance of those species within a local habitat. So in this case, within a lake or within a fish's gut. And I'll be talking about beta diversity, which is sort of a pairwise comparison of two communities that sort of quantifies the differences in species in those communities and the relative abundances of those communities. And the main point here is that alpha and beta diversity are often connected. So if alpha diversity is higher, then that says something about beta diversity in a lot of ways, because often higher alpha diversity might mean lower beta diversity because there's either greater sort of similarity in selection between hosts or environments, or there's sort of a higher level of dispersal between those two hosts or environments. So what do we find? We find this sort of parallel pattern across both systems where richness is lower, so we have lower alpha diversity in the hosts. So what I'm showing you here is a box plot of ASV richness, so the number of bacterial species we see in either a fish microbiome or a lake in both systems. And we see this sort of similar pattern where there's more diversity in lakes than in fish guts. And we also see that there's sort of higher beta diversity. So what I'm showing you is sort of within lake beta diversity. So on average in the same lake, how different are two fish or how different are two water samples within a lake? And what we see is that there's higher beta diversity in these fish compared to these lakes. What that could sort of suggest is that there's sort of stronger selection, sort of constraining diversity in these host microbiomes, or there's sort of dispersal limitation between hosts that is sort of elevating beta diversity and constraining alpha diversity. So this is pretty interesting that we see this sort of pattern, and I don't think it's really been shown in other papers before where we have this variation in diversity between these two community types. Cool. So what about the second question? Do hosts and free-living microbes display varying distance decay rates? And what I mean by distance decay is sort of this relationship where the similarity of two communities decreases as they get farther apart from one another. And this is really driven by two processes, either environmental heterogeneity between habitat patches that are far away from one another, sort of sort species based on sort of deterministic niche differences, or there's sort of random dispersal limitation between homogeneous habitat patches. So this is sort of the neutral theory, neutral paradigm. And we can quantify the strength of this relationship by just looking at the slope. And we can think about this as being the rate of spatial turnover. And by examining this relationship, we can ask how sensitive these communities are to spatial and environmental gradients. Cool. So we could sort of come up with some hypotheses and expectations that we could test. And so one sort of expectation could be that maybe we see sort of a stronger spatial turnover in the hosts compared to free living communities. And that's sort of what's shown by this conceptual figure here, where there's a more strong slope in the hosts compared to the environmental communities. And I thought maybe that this could be driven by dispersal limitation. So maybe it's harder or sort of a neutral dispersal limitation, where it's maybe harder for a microbe within a host in a fish gut to sort of disperse from one host to in a lake to another host in a faraway lake, right? Which makes sense. Alternatively, we could see this opposite pattern, or maybe sort of there's host filtering or sort of like non-neutral dispersal limitation or species sorting that would sort of limit turnover between hosts across space. So we'd see sort of a lower slope in hosts compared to the environmental communities. And the idea behind this is that we have the same host or same environment and different systems selecting for the same microbes in their guts. And what do we find? We find that water microbiomes have higher distance decay rates and greater sensitivity to spatial gradients. And so this is going to be a lot of data that I'm just going to throw out right now. But what we have here is the distance decay relationship. So On the x-axis is how far apart two communities are from one another. So either two hosts or two lakes, and sort of their similarity, right? So how similar are two communities to one another? And we can plot these, and what we can see is that there's a ton of variation in the fish communities and much less in the lake communities. And a stronger slope in the lake communities compared to the fish communities, and a stronger Mantel test, which is sort of an index of how much the strength of the relationship, essentially. And what I want to point out here, and the main takeaway from this, is that we see a pattern where sort of the beta diversity, which we could think of as community similarity, in individual fish is sort of like less related to space than in lakes, right? So there's sort of dispersal limitation and sort of host selection on the individual host level, as opposed to sort of as a function of what lake they're in. And we see sort of the opposite pattern here in these lakes where there's sort of dispersal limitation, but it's related to space. So there's less turnover within lakes, more turnover across lakes that are far apart from one another. So this is in the Sierra Nevadas, and I'm curious to see what we see in the Vancouver Island lakes. And we see the same exact pattern. So sort of a stronger relationship in brook trout compared to stickleback, but a sort of similar overall broad pattern where there's a ton of variation in these hosts. So a fish can be as different or as similar in the same lake than two fish in very far apart lakes. So this sort of reveals this idea that the composition of microbiomes in fish is sort of less related to space than in lakes. Cool. So we sort of talk about these patterns and these observational patterns, but it's really hard to parse out whether it's sort of neutral processes or deterministic processes are shaping these communities. And so an approach is to really sort of fit a neutral model to examine how much variation is explained by neutral processes. And to do that, we can plot sort of the occurrence frequency. So if we take all of our microbes and pull them all into one big meta community, we can ask, okay, how often does a microbe occur in this community? So essentially, if you have 100 fish, what proportion of fish does this taxa occur in? And we can plot that as a function of the mean relative abundance. So how abundant, how dominant is this taxa in these systems? And what the neutral expectation would be is that if all of these taxa have the same dispersal ability and same fitness, then we'd expect there to be increasing frequency as these taxa become more abundant. So really just by chance, if you're more abundant, you're going to be able to disperse to more habitats and more hosts. And so this is a lot to look at, but basically what we can do is each individual point here sort of denotes an individual taxa, and we can plot sort of its mean relative abundance or its occurrence frequency as a function of its mean relative abundance. And we can fit this model to sort of ask what taxa deviate from neutral expectation and how much variation in this is explained by neutrality. And we can sort of split these taxa into different groups. And so if you're within the confidence interval of this model, we could say that you're sort of putatively neutral. So you meet this neutral expectation. If you're above this line, then you're either sort of under positive selection or you're really good at dispersing. And what this means is that you occur more frequently than one would expect based on your abundance. In contrast, if you're below this line, then maybe you're under sort of negative selection or you're not as good at dispersing. And this model also sort of estimates this M coefficient, which is this migration rate estimate. So how much dispersal is there in these communities? Hopefully this is clear. If you have a question, please feel free to ask. Cool. So we fit these models to our data. And what we see is in these lake communities, we see these pretty similar patterns where a decent amount of variation is explained by this neutral model. And what I want to note here that's going to be highly in contrast to fish is that we see this pretty even distribution of microbes across the full range of frequencies. So there's a good amount of taxa that are found across many lakes. But in contrast, we see that there's sort of more non-neutral processes going on in these fish. So this model, the fit isn't as good, and this sort of M migration rate is much lower in these fish. So there's sort of stronger dispersal limitation between fish. I think I paused here. Can y'all hear me okay? My okay. Sweet.
Yep. Yeah, you're
Sweet.
fine, Josh.
Sweet. I froze for a little bit, so I was just making sure. Um, yeah, so there's sort of more non-neutral processes sort of driving assembly here. And what I want to point out here is that there's really these sort of above prediction points. And just as a reminder, these taxa occur more frequently than what you would expect based on their abundance. And so potentially these taxa are sort of more widely dispersed than you'd expect, but they're really never able to outcompete the other taxa in the community and become highly abundant. So I thought that was sort of interesting that we see this sort of parallel pattern across systems. And to just sort of emphasize this parallel pattern, I wanted to show you all the plots together, and we can see that the lake systems look similar to one another, and the fish look similar to one another. And so this also sort of gets at this idea of the core microbiome. And so the core microbiome is this idea that hosts or environments sort of have similar microbes as a function of host phylogeny. And we can see that there's a good evidence for that in these lakes, but not so much in these fish. There's not a lot of taxa that are really highly occurrent across all of the fish. Cool. Hopefully that made some sense. And so what are the key takeaways of this first project here? One, sort of the take-home message is really that these free-living and host-associated microbiomes are differentially affected by neutral and deterministic processes. And this variation in processes driving assembly sort of leads to variation in their biogeography. And we see this through sort of different diversity and distance decay patterns. And we also see this in how there's differences in neutrality shaping these communities, where in fish hosts, there's sort of stronger divergent selection or sort of non-neutral dispersal limitation between hosts. And it's pretty cool that we see parallel results from two distinct systems. And so these two fish are highly evolutionary divergent from one another. They live thousands of kilometers away from one another, and we still see sort of similar patterns. And the same thing for these lakes. So this sort of has implications for processes happening at sort of higher order levels. Sweet. Cool. So this is the first project I'm talking about. And this sort of leads to this second question. Does host community diversity and composition affect microbiomes? So we were sort of interested in this question based on this paper from Burns, where they found that whenever hosts are reared with one another, it increases their microbiome diversity. So what they did is they took zebrafish, a wild type zebrafish, and then they knocked out an immune gene and this other genotype using CRISPR, and they reared these fish in sort of three different conditions. They either reared them by themselves in sort of solitary conditions, and then they sort of reared them with multiple hosts, but sort of separated them by genotype. And then they sort of did another rearing where they mixed the genotypes together. And they predicted that solitary hosts would have the lowest diversity, separated hosts would have sort of medium diversity. And then when you mix two genotypes together, they'd have the highest diversity because in theory, two genotypes have different microbiomes. If they're dispersing microbes to one another, then they're shaping each other's microbiomes and sort of driving diversity higher. And what they found was that, yes, solitary individuals have lower diversity, but there was no sort of difference between these separated and mixed groups. So sort of genotype level differences didn't seem to drive higher microbiome diversity in this experiment. But I started thinking about, well, how might interspecific variation between hosts drive these patterns? And so this goes back to this host by microbiome by environment feedback I was talking about earlier, where essentially we have in this picture here, two different species of zooplankton sort of dispersing different microbes into the environment when they're separated from one another. So what happens whenever you rear them in sort of the same system? And so we have greater genetic divergence between these hosts, and we can sort of think about them as heterogeneous habitat patches, and again, the sort of feedback that I mentioned earlier. So specific questions, how does this affect their microbiomes? So the diversity and composition of hosts. And then sort of a higher order question that I don't have data for yet, but sort of how could this subsequent structure of these microbiomes affect ecosystem functioning, which in this case being host fitness. So what we did to get at these questions is we took three different species or three different sort of genera of zooplankton from a single lake in the Sierra Nevadas, and we manipulated the diversity and composition of uh, aquatic mesocosms to ask how the diversity and composition of hosts affects microbiomes. So just to sort of establish these taxa, these zooplankton taxa have distinct microbiomes. So this is from a paper that's currently in review that we wrote, where we sort of looked at the microbes in the field and sort of compared them between different hosts. And what I want to point out is that we see this strong separation between different species of hosts. So what we did is we took, in theory, what we tried to do is we took these large mesocosms, we seeded them with zooplankton and sort of a gradient of diversity. So we had three different species, Daphnia, cyclopoid, and calanoid copepods, and then we reared them in single species treatments and sort of pairwise of the different combinations, and then sort of a treatment of all three together. And then additionally, we had tanks with no zooplankton in them whatsoever. And then we wanted to sort of think about how this feedback could affect the fitness of the hosts. And so we took Daphnia from either single species Daphnia tanks or sort of multiple species Daphnia tanks and reared them in different environmental conditions and looked at sort of how that could change the fitness. So sort of the growth rate, population change, and sort of biomass of those hosts. And so the prediction is maybe that we see this increase in microbial community richness as a function of host community richness. So the idea is that sort of interspecific microbiome dispersal sort of will drive higher host and environmental microbiome diversity. So we're thinking about how this feedbacks and not just the host, but also in the environment. Cool. So unfortunately, our experiment didn't turn out how we wanted it to. So what I'm showing you here is sort of a relative abundance plot of our tanks, our treatments over three different time periods. And the idea was we were going to have sort of a diverse or richness gradient where we have this really clean separation by species. And this would be a lot to sort of go through this whole figure. But essentially what we see here, what I want to point out is that Daphnia became really dominant across all of these treatments. And so what happened is when we were sort of separating out sort of zooplankton into different treatments, there was some contamination of Daphnia. So either sort of eggs or really small Daphnia that got into sort of the wrong tanks. And so we didn't really get this clean richness gradient that we were hoping for. And so we can't really test this hypothesis of how community richness could be shaping microbiome richness. So that's unfortunately not going to be able to happen. However, we still have sort of these experimental gradients over time. And so we could look at sort of host Shannon diversity as sort of a continuous metric of diversity rather than richness. And this really changes quite rapidly over time as the dynamics of the host communities change. We also saw this pattern where the abundance of hosts changed in these tanks over time. And so we could sort of use these two experimental gradients that we've generated sort of unintentionally to sort of ask a couple of different questions. So instead of looking at richness, we could ask, how does Shannon diversity and abundance affect microbiome diversity? And we could sort of predict that maybe a more diverse host community could drive higher microbiome diversity, and a more abundant host community could drive higher microbiome diversity. So just looking at our microbiomes, what I'm showing you here is a PCOA plot. So each point here represents a microbiome from either calanoid copepods, cyclopoid copepods, Daphnia, or tank water. And what we can see is that there's sort of strong variation between species, which is what we're hoping for. And we can see that a considerable amount of variation is explained by this, so 31%. And there's some variation explained by time point and some by treatment. And treatment's a little bit, that metric is a little flawed because the treatments didn't exactly stay the same over time. But we can still test these different questions about how Shannon diversity and abundance affect microbiomes. So first, how does host diversity affect water microbiome diversity? We really don't see a relationship. There's a lot of variation. It looks like there might be something, but it's not significant. In contrast, we see that there's sort of varying responses to host diversity for the host. So what I'm showing you here, I know this is a lot of data to look at, but we'll walk through it, is we're looking at sort of the diversity of microbiomes within different hosts, so calanoids, cyclopoids, and Daphnia, as a function of the diversity of hosts in the whole community. So we predicted that we would get more Shannon diversity in the microbiome as a function of diversity in the hosts. We actually see sort of significant relationships, but there's different responses between the different hosts. And I just got this data back about 2 weeks ago, so sort of curious about what could be driving this pattern. And one thought is that we see sort of variation in the abundance of these different hosts. So I'll be sort of digging deeper in to see what could be driving these patterns. But the main point here is that hosts are sensitive to zooplankton diversity in their microbiomes, but the water is not. But so now we're looking at abundance. And what we can see is sort of a similar plots where we have each sort of sample type and predicting its Shannon diversity as a function of zooplankton abundance here. What we can see is there's not really any relationships. So seems like host diversity could be driving patterns, but not zooplankton abundance. But how does host beta diversity sort of interact with this here? And so what this plot here is asking is sort of how host community composition influences microbiomes. So if there's sort of two host communities that are really different from one another, does that mean that the microbiomes in those host communities will be really different from one another? So sort of does host beta diversity drive microbiome beta diversity? And what we find is that microbiomes are structured by host beta diversity. So this is, again, a lot of data with a lot of variance I'm showing you here. But on the x-axis, we have host community dissimilarity. So again, how different are two communities of hosts from one another from two environments, two tanks? And then sort of, is that related to how different microbiomes are for calanoids? So this is comparing calanoids to one another from different sort of communities, cyclopoids, Daphnia, and water. What we can see is this sort of similar pattern across species where there's stronger effects on plankton microbiomes, but sort of minimal effects on water microbiomes. So what this sort of suggests is that the structure of the host communities in the environment and interactions between hosts can sort of scale up to influence the microbiomes of those hosts. So what are the key takeaways from this sort of section? One, host community dynamics and interactions could be sort of an overlooked process altering microbiome composition and diversity. And this potentially could scale up to influence host fitness and ecosystem functioning, which I'll sort of be talking about a little bit more in the next section. And we see these sort of species-specific responses to host diversity. And this could be sort of connected to individual host abundance. So they show sort of different relationships based on how abundant they are in the communities. And we sort of see no effect on water microbiomes. And so how is this dispersal really working? We'd think that if there's sort of hosts with different microbiomes interacting and dispersing microbes from one another, we would see sort of a signal of that in the water. But that's not really happening. So it's kind of an interesting result here. And then also sort of host community beta diversity influence host microbiomes. Oh, let me back up a little bit. So future directions for this project is really asking about, could this scale up to influence host fitness? And so this is getting back to this inoculation experiment. So if hosts are interacting and shaping the environment of microbes around them, which we didn't really find, could this dispersal sort of affect hosts? And so this is sort of the inoculation of experiment of inoculating Daphnia and the water from sort of different host diversity treatments and sort of evaluating fitness proxies. Sweet. So now I'm going to move to my final section of the talk, which really asks, what are the consequences of microbiome divergence amongst ecologically and evolutionary distinct species pairs for host fitness and ecosystem functioning? That's a really big mouthful, but I'll be breaking this down. And I'll be aiming to answer this question using sort of benthic limnetic pairs of stickleback. So I'm really interested in understanding the effects of microbiome phylosymbioses. And really, this is this idea of there's this host microbiome coevolution, where we see that hosts sort of select for microbes that are very particular as a function of their species. So as two species become more phylogenetically distant from one another, their microbiomes become more phylogenetic, or more phylogenetically and more compositionally distant from one another. And conceivably, the effects of this relationship could sort of influence the fitness of hosts. And so we could see sort of a gradient where there's some sort of peak in host fitness across different microbiome diversity and compositions. We also know that the fitness of hosts influences ecosystems, especially when hosts are sort of keystone species or ecosystem engineers that affect the functioning of ecosystems. And so really want to interested in thinking about is sort of the indirect effects of this host microbe coevolution on ecosystems. So can sort of changes in host microbiome interactions sort of influence host fitness, which could indirectly scale up and affect ecosystems. So to get at this question, I want to use three-spine stickleback as an eco-evo model. And if y'all aren't familiar, these are fish that are a well-known model in evolutionary ecology. So ancestrally, they are marine. And then after the last glacial period, they've repeatedly colonized these freshwater habitats pretty globally in the Northern Hemisphere. And when they've colonized these environments, they've evolved these sort of parallel traits independently from one another. So it's a really good system for understanding how species sort of adapt to new environments and evolve independently from one another according to sort of environmental selection. And a really interesting sort of process and system nestled within stickleback is this benthic limnetic species pairs. And so I think in five of these lakes in Vancouver Island, there's this sort of sympatric speciation between benthic and limnetic stickleback. So benthic stickleback and limnetic stickleback have very different morphologies. They have different ecological traits. They have different diets, different genetics. And so benthic stickleback feed in sort of the littoral habitat, which is sort of the shore water environment, sort of shallow water, and they feed on mostly macroinvertebrates. And limnetic stickleback live in sort of the open water environment and feed on zooplankton prey. And we also see that these different ecotypes of stickleback sort of affect ecosystems in different ways. So this is sort of evolutionary diversification that has consequences for the functioning of ecosystems. And we also know that there's sort of differences in the microbiomes of these ecotypes. So we get sort of this morphological, evolutionary, and microbiome divergence between these two ecotypes. So what I'm interested in asking and sort of proposed to SSE was sort of asking, what are the consequences of this divergence for host fitness and ecosystem function? And to get at this, I proposed an experiment where you sort of take microbiomes from benthic or limnetic stickleback and transplant them into benthic or limnetic stickleback in sort of a pairwise fashion. And so what this is showing here is sort of that, where we have benthic stickleback being red, limnetic stickleback being blue, and we take the donors, take their guts, introduce them to recipient stickleback to sort of introduce a benthic stickleback to a benthic or benthic microbiome to a benthic stickleback or limnetic microbiome to a benthic stickleback, and sort of the same for limnetic recipients. So sort of transplant these microbiomes and then put them into experimental mesocosms. And so the setup would be you would take these four different treatments, put them into experimental mesocosms with zooplankton prey, and then also sort of have an additional fish list control and examine sort of the potential ecosystem effects of these stickleback and whether if you get the microbiome from the same ecotype, that influences your fitness on the right, so sort of your growth rate or body condition, or sort of your influence on the environment around you. So sort of driving changes in productivity, so trophic cascades, or sort of altering the prey density. So in this scenario, maybe we'd expect sort of if you got a microbiome from a different ecotype, you would be sort of have lower fitness and have different effects on the ecosystem than if you got the microbiome from your same ecotype. Sweet. So this is the experiment I proposed and really hope to do soon, but there's a lot of groundwork that we have to sort of get through to get this experiment going. And so to get at this, we did a quick little pilot experiment to sort of examine how we would go about doing this, especially in terms of the microbiome transplants. So what we did is we sort of had the same recipient population. So this intermediate host, we called it. So there's sort of a benthic and limnetic axis, and there's a lot of populations that lie within that axis. So we took a population that's sort of intermediate and we introduced them to antibiotic cocktail overnight in sort of sterile water. And then after treating them with antibiotics, we introduced guts from either benthic stickleback, limnetic stickleback, or sort of the same population. And then finally, we had sort of an antibiotic only control to sort of examine independently the effects of antibiotics. So really, we're just trying to get at how well does this transplant work, and do antibiotics have an effect in general, especially on diversity? And when we see the sort of minimal and sort of varying effects of the transplant and also antibiotics. So on the y-axis, we have sort of the percentage of the microbiome in the recipients that came from the donor. So sort of like what proportion of microbes in a recipient fish is from the donor. And on the x-axis, we have the different treatments. So if they were transplanted with a benthic microbiome, whether or the same intermediate sort of same population or limnetics. And what we see is that there's variation, right? So for the limnetics, it was really successful, but for the benthic and the intermediate, it was less successful. So there's sort of varying success of this. And we really didn't see an effect of antibiotics, which I found pretty surprising. So what I'm showing you here is Shannon diversity of fish microbiomes on the y-axis and the different sort of donor treatments on the x-axis. What we see is there's really no difference between treatments. And I really was curious to see if whether there'd be a difference in the antibiotics, and we really don't see an effect. And then finally, we sort of looked at the structure of these communities and whether sort of treatment could be driving the composition of these communities. And we really don't see that. So this is a PCOA plot. It's a lot to look at, but the primary thing that I want to get across is that treatment only explained 1% of the variation in community structure in these fish microbiomes from the experiment. And it was marginally significant at 0.028. So what are sort of the future directions for looking at this question and what I propose to SSE? So first, we've got to do some more antibiotics treatments trials. So sort of titrate to examine and sort of get the correct dosage of antibiotics. And really the challenge seems to be is getting microbial inocula to colonize host guts. And so I probably should have been more specific about how we did this, but it's pretty challenging to sort of seed microbes in fish guts. And what we did is we sort of reared fish in these small little tanks of sterile water, and we introduced the guts of whatever we wanted to donate to them into the water and let them sort of be in there overnight. So really want to think about different ways to optimize this transplant. So there are some methods out there to rear germ-free stickleback. And so this might potentially make it easier for donor microbes to colonize the guts of hosts if the antibiotics aren't quite doing their job and getting the getting the microbes to decrease. Or we could sort of do a manual gut transplant to sort of syringe inocula into the guts of fish, which seems a little more challenging, but potentially an option. And really, I think the idea behind this experiment would be the first to sort of connect evolutionary divergence between hosts and their microbiomes, the sort of ecosystem functioning and also host fitness. Cool. So what is the summary of the whole talk? One, we found that hosts and environments have really different microbial communities and they display divergent biogeographic patterns. And this is sort of driven by variation in the processes driving assembly. Two, this is a lot of words, I'm realizing that now, but host community dynamics and interactions can alter microbiome composition and diversity. And this is potentially sort of an overlooked mechanism influencing microbiomes and potentially host fitness. And finally, I want to try to get at sort of more the consequences of microbiomes. And so this is where this project, this experiment of examining differences in benthic and limnetic sticklebacks and how that affects host fitness and by extension ecosystems. And so I think this framework is really important for microbiome studies moving forward. So with that being said, I'd like to acknowledge all these amazing people who have been very helpful throughout my PhD, in particular, Diana Rennison's lab, John's lab, and all of my collaborators and my funders. So thank you all for listening. And if you have any questions, I'd love to hear them.
Great, wonderful. Thank you so much, Josh.
No worries. Thank you.
So, for those of you who have questions, there's a couple ways we can do this. You're welcome to post questions in the chat, and I'll try and keep my eye on that. Or you can raise your hand with the React button, and we can we can try to answer questions that way. Robert, if you have other ideas.
That's all great. People can turn their cameras on if they want or type in the chat.
hmm So I can start maybe. Um, and Josh, let me just tell you that I am only just starting to dabble in the microbiome, so
Mhm.
forgive my yeah if I mess something up with my question. But um,
Worries.
so with your first project, when you were looking at the trout and then the sticklebacks in their
Mhm.
respective lake environments, um, I was wondering when you were seeing that there was a fair bit of difference between individual fish, even when they were in the
Mhm.
same lake, in diversity. Um, did you still have a lot of overlap among the specific microbes that were present in the microbiome, or did you have some fish that
Mhm.
had like totally different actual like microbes, even if the
Mhm.
proportion of the different microbes was somewhat similar? I don't know. Does that make sense?
Yeah, so sort of like a presence absence versus relative abundance kind of question.
Yeah,
Yeah,
yeah.
yeah, yeah. So um, a lot of the variance we see is sort of like on the relative abundance scale, but also both, I guess. It's just so much variation. But one
Mhm.
thing I can say that makes it a little more definitive for you is like in these microbiome studies, we look at sort of different metrics of how different microbiomes are from one another. So sort of like just on an ASV level, which is like another word for a microbial species,
Mhm.
how different are they in numbers and how different are they in sort of presence or absence? And those that seems to have a lot more variation. And we
Okay.
could also sort of look at the phylogeny of those ASVs and be like, okay, how similar in phylogeny are sort of the microbiomes of two fish? And we see
Yeah.
there's sort of less variation in phylogeny than in sort of just looking at quantitatively ASV level
Mhm.
differences. And so there
Okay.
seems to be sort of host selection, but the specific ASVs within a host can be widely varying between two hosts within the same lake.
Huh. Okay.
Does that make sense? I don't know if that quite answered your question.
No, I think you did. I think you did. Yeah.
Yeah. Okay.
Thank-
Great.
mhm.
Thank you for the question.
And then it looks like we have a question in the chat um,
Cool.
from Jonathan Shurin. So, for the fish microbiomes, the
Hmm.
neutral and non-neutral microbes seemed bimodally distributed. Do you
Hmm.
know if the over-distributed ones are evolutionarily distinct from the neutral ones?
Yes, John. Yeah. Let me show y'all. So, I had this figure. If you don't mind, I'm gonna have to hop out of this to show y'all. Um, Sort of a PCOA plot. Okay. So I'll sort of explain this figure here. So we have hopefully it doesn't hide it. It did hide it. Dang. So we have these different microbes, whether they're above, neutral, or below. And he's sort of asking if these sort of over distributed ones are different from the neutral ones. And so what we can do is we could sort of make these different communities, right? So we can sort of group above brook trout together, above stickleback together, above Sierra Nevadas together, and then the neutral and then the below, etc. And we could sort of calculate, okay, how different are these communities from one another and plot that. So each different color here represents whether what sample type they're from, and the shape represents sort of like what putative selective part you're in. So are you neutral? Are you above the line? Are you below the line? And this is a PCOA, which sort of looks at phylogenetically how similar are those taxa to one another. And it's kind of hard to see, but we can see that, like, for example, in fish, the below taxa are really similar to one another phylogenetically. And in water, the below are really similar. And the neutral ones are actually more similar to the above than the belows. Is this making sense? And we can sort of look at, okay, how much variation is explained by sort of what selective regime they're in. And 21% of the variance is explained, which is fairly decent for microbiome studies. So to get back to John's question, yeah, they are. We do see phylogenetic and sort of taxonomic differences between those different groups from the neutral model. Hopefully that made sense.
So then do
Yeah.
you think that those phylogenetic similarities within the different groups, do you think that's linked to their function, what function they
Yeah. Yeah. So with this is all 16S data, which makes it hard to look at function. But the goal is to sort of do more metagenomic data, metagenomic studies to sort of follow up and look at whether there's different functions associated with these different groups. And so I could think maybe you would expect ones that are sort of above neutrality, so they're sort of they occur more frequently than what you'd expect based on their abundance. Maybe there's some sort of functions that are related to dispersal that could be allowing them to disperse really easily between hosts, but they're not able to become very abundant in those communities potentially because there's often this trade-off between dispersal ability and sort of competitive ability and colonization ability. So that's a potential thing to look at. And that's something that I really want to do with sort of these studies is to look at, and it's something that Andy and Diana are looking at, is sort of looking at, sorry, it's a lot of moving around, sort of looking at differences in microbiome function between benthic and limnetic stickleback, and if those functions are sort of parallel between the different populations. And so it would be cool. And Diana's paper sort of showed that there was differences in function, but this was sort of on a 16S, you
Mhm.
know, meta barcoding approach, which is sort of less precise for looking at function compared to a metagenomic approach.
Yeah, interesting. Robert, I think I interrupted you.
Yeah, we hopped in at the same time, I think. Um, I just wanted to ask because it sounded like the difference between the water. Actually, Josh, first I want to say, yeah, I don't have a background in the microbiome or ecology, but you did a good job teaching
Sorry,
about
I hope
each.
it
No,
was a
no,
lot of data and pretty, yeah.
no, I'm saying the slides beforehand that sort of explained the coming analyses were very helpful, and you did a good
Okay, good.
job going through the example. So there was no need for me to interrupt because I was clear on a lot of them.
Great. I'm happy to
So,
hear that.
but I just wanted to ask, yeah, so good job. But I wanted to ask, so it sounded like you were surprised that the water and the inside the fish gut were as different as they were. Like
Mhm,
initially going into it, you thought it was going to be a little more similar. Like, so
mhm.
do these microbes they don't have part of their how are they living? Part of their life cycle is not in the water, and then part of it's in the gut, or
Yeah.
when they're dispersing between fish, because the dispersal was uh what was less obvious in the fish between the different lakes than the water. So they
Mhm.
are dispersing. So I'm assuming they disperse through the water. So they must be in the
Yeah.
water sometimes, and yet the
Yeah.
water is so different. So
Yeah. Robert, this is a great
yeah.
question. And we see this sort of same pattern in the plankton where like
Huh.
the plankton sort of seem to affect each other's microbiomes, but we don't really see that dispersal in the water. And so I did
Yeah.
expect them to look a little more similar. And I think the idea was going back to like, okay, there must be some dispersal through the water, but also sort of the environment within the gut of a fish is so different from the surrounding water.
That's
Like
a good point.
pH is
Yeah.
different.
Hmm.
It's, there's no oxygen. It's really highly differentiated. And so I think what I'm thinking about with your question is sort of like, okay, where are these microbes coming from? Is it sort of vertical
Hmm. Yeah.
transmission? Like when a fish lays their eggs and it's fertilized, is there some microbes in there that really colonize the host, and that's primarily where we get the composition of the microbiomes? Or another potential possibility is that um, this dispersal is so transient that it's hard to capture whenever we sample it,
I see.
right?
Yeah.
Or these microbes that are dispersing between hosts are in such low abundance that it's hard to capture them in our analyses. Like, for example, you'd have to sequence super, super deep, tons and tons of reads to find those reads that match to the microbes that are in hosts. And so that's something that we've been thinking about a little bit. Sort of like, okay, how is this really happening? Right? Like, could be sort of multiple of those things could be sort of explaining this at once.
Yeah. Thanks for the great answer. I want to get to the last question there's one more
Cool.
question in the chat, I think. But yeah, I had been thinking vertical transmission, but then when I saw that lack of a relationship between uh fish at a distance, sorry, I'm not doing a good job of explaining the
No,
joke,
I think you're, yeah,
but
hopefully I
that
explained it well enough.
that made me question vertical transmission and then wonder, okay, well, where is it in the water? But anyway, there's one more question there.
Totally.
Yeah.
Mhm. Yep. So our last question. Nice talk. I'm wondering if the developmental stages of fish and environmental temperatures of the water could have an impact on microbiome communities.
Sweet. That's a great question. So I think there have been quite a few studies looking at sort of development and microbiomes in sort of a more Evo Devo way, especially with zebrafish. And I think there are some sort of successional stages and the composition of the microbiome sort of over development. I'm not as familiar with those studies, but yeah, that's sort of an interesting avenue to sort of look at how development sort of interacts with the microbiome. I know there's a lot of sort of questions about that and some studies on that. But yeah, to my knowledge, yeah, there seems to be sort of a successional stage in a microbiome over development. And as far as your environmental temperature question, so I didn't talk about this, but one of my chapters in my dissertation sort of asks this exact question. So how could sort of variation in temperature influence microbiomes in fish, and could that sort of influence growth and fitness? And the answer is we don't really know. In my study, there didn't seem to be a strong effect of temperature. But I also used a host that could just be more tolerant. And there's some other studies that seem to show that there is a strong effect of temperature on microbiome composition, and that microbiomes could be an important sort of source of adaptation to environmental change, especially with like, I think there's a study that came out pretty recently. I can't remember if it was Nature and Climate Change or what it was, but it was sort of on the microbiomes of frogs and sort of how changes in temperature and climate change could be influencing their interactions with their microbiomes and how microbiomes could help them adapt. So I think the answer to your question on the environmental temperature thing is that it probably is widely varying between hosts. It really depends on their physiology and relationships with their microbiomes.
Well, wonderful. Thank you so much, Josh. This has been super interesting, and yeah, thank you for speaking to us.
Thank you so much. I really appreciate the opportunity to talk to you all. And I'm looking forward to attending more of these talks. So thank you.
Okay, great. We'll see you guys next week for the next Greg seminar here. Thanks a lot,
Have a good
everybody.
one.
Thanks, Josh. Thanks, Amber. See you
Thank you.
guys.